Alright, my name is Robert Higgins and this is my brother Harley Higgins. We're going to be talking about DNA and transcription and translation. So here I have my DNA strands, and we've got two of them here. And I've with DNA there's four letters that make them up. There's A, G, T, and C. A is color coded as green and it's 2-ring base. Yellow is T and it is 1-ring base. Red is 2-ring base and it matches up with orange with 1-ring base. So I have this sequence all the way down here making this strand up. So we need to make protein. So when this happens we need to copy a strand, which we're going to copy the left one. And how we do that is with RNAP. This is an enzyme that's going to come down the strands and it's going to break them apart temporarily. So as it's coming down, it's taking them apart and it's reading, which we're saying is the left side. And as it's doing that, it's pumping out the RNA sequence. So as it moves down and it's finished copying, the right side is actually identical to the RNA. Now the only differences in the RNA sequence is that yellow has been replaced with white. So that's T is now U, which is uracil. So now that it's been copied, we can move the DNA out of the way. Now we have our RNA strand. The RNA strand will move out of the nucleus in the cell and it's going to find a ribosome and it's going to enter it. The ribosome is going to read the code and we'll start, actually also while outside the ribosome and the nucleus are amino acids floating around. So this is our ribosome here. We'll do it this way so the camera can see. So as it's reading, it reads them in 3, the letters by 3, which are the codons, which identify which amino acid it needs. So our first colors are white, green, green, which is UAA. So we've got our first one here, methionine, which is a little egg carton. And this is our first one. It's floating around. Bam. There's our first one. So we'll get this ready. Okay. And so the ribosome again pumps out the next 3. 1, 2, 3. We've got white, red, red, which is UGG, and it's glycine. So again, it was coming around, plopped on, and now we can connect up with the next one, which is leading to making our protein. Alright, that fell off, oh well. Okay, so that's our first 2 there. So we just did UGG. Now our next strand, or our next sequence codon is green UU. So, or green white, which is AUU. So we have that, it's leucine. So again it would come around, plop on, there's our next set. And then again our next one, it reads out. It is white, red, which is UGG. And we've already had that one go through, so we have another one. It comes around, touches on, and it assembles there as they're coming out of the ribosome. Next one is orange, green, white. So that's CAU. So come around, and we've got another one. Okay. Now our next set is red, red, white, so GGU, and that one is an interesting name. And let's get at the next one, which is green, orange, and white, which is ACU. So as the ribosome's reading them all, it's printing out the appropriate amino acid to create the protein. And protein can be turned into whatever it needs to be, different tissues, and whatever the body needs the protein for. So after the DNA has been coded and been ripped apart by the RNA-P, it's created the RNA strand, which then leaves the nucleus, finds the ribosome, which reads it, and attaches which takes the appropriate amino acids and creates the protein that's needed for the human body.